Hello and welcome to my presentation for the Bio 182 Lab Final Project. In this presentation, I will be comparing a news article and a scientific paper over the topic of snake evolution and will later determine if the news article is accurate in terms of explaining the experiment and results from the scientific paper. The news article I found on the New York Times paper is titled How Boa Constrictors Breathe While Squeezing Their Life Out of Their Prey. This was written and edited by Sam Jones on March 24, 2022. I will now summarize the major points given in this article. This news article begins by explaining the background behind boa constrictor behavior and diet. Boa constrictors squeeze their prey to stop blood flow for quick and easy consumption. They will eat opossums, rats, some of their own snake relatives, capybaras, and in a few rare instances, humans. Boas typically squeeze their prey for 10 to 45 minutes depending on the size and build. But if boas constrict by also squeezing their whole body, there has to be some way for the boas to breathe while doing so. This was the question posed by Dr. Elizabeth Brainerd and her colleagues. The article introduces Dr. Brainerd and her fellow researchers and how this experiment and findings were published in the Journal of Experimental Biology. The article notes that humans breathe with their diaphragm, birds breathe with their small air sacs, but boa constrictors have to fully depend on their ribs and long physique to breathe. It is thought that they slowly expand a section of the rib cage to create pressure change and let air in. The rib cages also expand when ingesting their prey. With all of this in mind, it is puzzling on how boas are able to breathe and eat at the same time. The article then continues to sum up the experiment and results. The researchers hypothesized that boas are only expanding one specific region of their rib cage at a time. In order to test this, they made a visualization of a boa's rib cage using 3D X-ray models and implanted metal markers inside anesthetized snakes for imaging. They were able to take multiple images and videos with the X-ray. To simulate constriction, they applied a blood pressure cuff to the subjects for restricted rib movement. After analysis, the researchers found that in normal condition, boas breathe using the upper third portion of their rib cage. When this section constricts, the lower portion of the rib cage expands and takes in air. As quoted by Dr. Capano, one of the researchers, the snake just turns off a section of the rib cage and then turns on another section. Figure 1 to the right is just a picture of a boa constrictor. All pictures are cited at the end of this presentation. The primary literature and scientific article is titled Modular Lung Ventilation in Boa Constrictor. This paper was written by Drs. John Capano, Scott Boback, Hannah Weller, Robert Sierri, Charles Zwemmer, and Elizabeth Brainerd. This was published in the Journal of Experimental Biology on March 24, 2022. The following summary will include the major points from the most important sections of this paper. The introduction of this paper begins with the background and purpose of the boa constrictor experiment. The researchers noted that the evolution behind the diversification of snakes are not entirely clear, but constriction behavior and highly kinetic skulls stand out the most to animal biologists. They also theorized that before their constriction behavior could evolve, the snakes had to have gotten around the lung ventilation constraints. Because of this, the potential for snakes to actively shift the location of lung ventilation is plausible. To test this, the study required data on airflow, muscle activation, and rib kinematics of the boa constrictor. Figure 2 displays the boa rib anatomy of interest. Image B shows expansion while A shows the ribs at rest. The muscle of interest here that the researchers test will be the levator costa muscle in image A. In conducting this experiment, 3 radio-opaque markers were placed into each rib of interest to produce 3D bone animations for all snake subjects. They collected X-ray video data from these markers to compare and analyze rib motion patterns throughout the body. The process using this technology was called the XROMM, which is short for X-ray reconstruction of moving morphology. To simulate constriction, the researchers applied a blood pressure cuff with consistent and controlled pressure and measured the physiological responses. Some of the XROMM trials used pneumographs to measure airflow. However, not all of the pneumograph data was reliable or consistent because the boas kept removing it. 2 of the trials had 2 boas subject to electromyography, or EMG for short, to determine if the levator costa muscle was capable of discrete muscle activation patterns for rib motions. Figure 3 shows what an EMG looks like on a human subject, and Figure 4 shows what a pneumograph looks like on a human subject. The researchers found that boas do actively adjust their trunk segments and ribs for lung ventilation while constriction takes place. In this case, their hypothesis was supported. The application of the blood pressure cuff forced the boas to rotate their ribs in the saccular region with no rotation in the vascular region. The researchers also found that boas were able to ventilate in the trunk segments near the constriction coil. EMG trials also revealed that boas could activate their levator costa muscle at will wherever they wish. As a result, they conclude that rib motions are a result of spatially discrete rib rotations and controlled muscle activation patterns. The researchers also concluded that modular lung ventilation may have been a necessity during snake evolution before constriction behavior could form. Figure 5 to the right shows the pneumograph and X-ray readings collected during the experiment. The statement to the left shows how to read these data. The news article was accurate in clearly stating the researchers' hypothesis and predictions. It also accurately summed up how the experiment consisted of blood pressure cuffs, X-ray imaging, and the radiomarkers. It also shared the major point of the researchers' conclusion that snakes basically turned one section off while the other is under pressure. Overall, I think it was accurate in summing up the major and most needed points from the experiment and results. For the inaccuracies, though, the news article did not mention the EMG and pneumograph trials and results. They also left out the discovery with the levator costa muscle and its activation pattern. It also did not explain any boa anatomy that might be needed to understand the study. Overall, I think the news article is more accurate than inaccurate. If you think about the audience difference between casual readers and scientists, it's understandable why the inaccuracies are there.